In this presentation, we introduce our project Parasitic Signals Multimodal Sonata for real-time interactive simulation of the SARS-CoV virus. This project began as an art project exploring the relationship in parasitism. From 2019 to 2023, this research evolved through collaboration between artists, scientists, and engineers, with scientific theory and data transformed into a simulation of the interaction between viruses and human molecules.
I must mention the first series of parasitic signals, which was developed from 2019 to 2021. During that period, we utilized the Borrelia bacteria known to cause Lyme disease in humans. While we were working on the first parasitic signals installation from 2019 to 2021, the pandemic emerged and research on the coronavirus accelerated across various fields of the study. COVID-19 has disrupted our healthcare, social, and economic environments. In spite of significant technological advancements made in managing this pandemic over the last 2 years, a substantial number of people are still infected and suffering from the virus. In fact, the relationship between humans and pathogens, along with their social and cultural impacts, has been the subject of study for quite some time. There's no need to mention historical examples like smallpox, typhus, and Spanish flu. Pathogens have been infecting human populations alongside social and cultural changes as well. However, this project aims to focus on the mutualistic relationship between parasites and humans by referencing scholars in contemporary philosophy and bioscience domains such as Richard Dawkins, Lynn Margulis, and Donna Haraway. In this sense, parasitism has been studied in this series of works as an interspecies relationship between humans and other species, examining various contexts in social, artistic, and scientific practices. Therefore, our research question aims to provide new perspectives on scientific discoveries in complex biosystems by creating interactive audiovisual simulations that depict the physical characteristics of the interactions between the coronavirus and human protein molecules. This approach not only aims to impart educational knowledge, but also to encourage conversations from diverse viewpoints encompassing art, science, and technology.
This project started with the scientific observation of the interaction between the SARS-CoV-2 virus and the human cell components. At UNS Kepler University links here, we operated high-speed atomic force microscopy and successfully recorded the first movie of the trimer of the SARS-CoV-2 virus. This breakthrough allowed us to characterize how the coronavirus can interact with the human protein inside the human body. Atomic force microscopy also applied to measure the mechanical interaction between the spike protein and the ACE2 human protein. For example, this graph shows a few examples of the interaction force data over time.
We started with a noisy AFM video of the COVID spike, used signal processing to get better imagery, labeled every single frame to explain the 3D dynamics, utilized a scientific protein model, and implemented dynamic rendering of COVID spikes in our custom C++ platform, including OpenGL and Allolib. A custom instance rendering method is implemented using OpenGL shading language to draw the dynamic and volumetric structure of the virus spikes. By filling out hundreds of thousands of vertices with transparent instances, the skeleton of the 3D model closely resembles the original AFM image. To elaborate on the relationship between viruses and humans from a nanoscopic perspective, we brought the infection model of coronavirus from the conducting zone of the airway to cellular membrane with ACE2 receptors. This model includes viruses, cells, and defensive systems like the mucous layer and ciliary hair above the cell surface. The ACE2 receptors are hidden by the mucociliary system and even move around the vast cell surface. We depict viruses and human components, referencing their scientific characteristics, including texture, color, and scale. The left figure shows the schematic image of components in the simulation with their actual sizes, and the right part shows the real-time rendered scenes. This project's narrative is driven by the infection mechanism, comprised of the appearance, diffusion, penetration, and binding for entry. Just like the actual infection progress, these stages can go back and forth or be accelerated or decelerated by the probability model and the real-time interactive components.
Audio in this project is a crucial modality that coherently represents the events of the SARS-CoV virus infecting a human cell. Every agent in the simulation emits spatialized audio, filling out the sonic landscape with dozens of real-time sound synthesizers. You are now looking at the audio spectrum of 4 minutes of the installation that demonstrates the various stages of infection. The sonic structure moves from the exposition to the development and at times recapitulates, reaching a climactic point and da capo over time. Throughout the sequence, this musical structure repeats and eventually flows to the end according to the probability model of the events. The formal structure is intentionally designed to be paired with the infection mechanism. We designed the real-time sonic landscape to sonify the data and the underlying process of infection, but also to compose a musical plot structure that aligns with the events in the simulation. As a result, the sonic landscape is filled with unique and dynamic patterns that are interconnected with the simulation, visualization, and narrative.
This figure shows the overall structure of the project's multimodal design. This logically explains how we depict invisible, inaudible, and untouchable nanoscale phenomena through real-time interactive multimodal experience. We designed the interface to invite the audience to participate in the audiovisual narrative. The interface incorporates two methods, a voice-based interface and a touch-based interface, allowing users to intuitively explore the dynamic biophysics of the coronavirus and human cells.
It was fortunate that we had the opportunity to showcase this work in a few public spaces where we were able to engage with a diverse audience from various backgrounds. Most people were genuinely surprised as they had not encountered the pandemic presented in such a comprehensive visual, sonic, and tactile environment before. Artists expressed a curiosity about the philosophical underpinnings and the aesthetic of the visual representations, while engineers inquired about software technology and interaction design.
Scientists and physicians provided feedback that not only improved the scientific realism of the project, but also highlighted different perspectives on imaging and understanding the coronavirus and its mechanism.
Nevertheless, this research primarily focuses on transforming data derived from the human-pathogen relationship into an artistic presentation of the scientific data. However, we also observed that since the pandemic, our research has promoted critical questions regarding power structures, ideologies, and the unequal impacts of the pandemic on various communities. These concerns include issues such as hate crimes, with a particular focus on those targeting Asians, as well as domestic violence and disparities in hardware availability within remote education systems. These intricate matters, closely tied to cultural and social backgrounds, can be integrated into our future endeavors as we coexist with the virus. For the forthcoming advancement of this research, we intend to address the societal aspects of our communities, expanding the scope of our research data to examine how pathogens can influence our society from the nano to macro level.